Yeah, thank you very much. So I am Dr. Prashanta Karunakar from Department of Biotechnology, Dayanand Sagar College of Engineering, Bangalore. So this is a work we did especially to in the pipeline of drug discovery, not to leave any candidates or the ligand molecules just based on the Vina score. So we want to extract the bonding interaction and then uh, to decide whether to consider a ligand or not. So our study is referred as Vinaliggen. So we have developed a method or a kind of a mini tool which helps us to generate the Ligplots, and we can retrieve the hydrogen bonds and hydrophobic interactions after performing the docking, that is protein ligand complexes. So here we have developed majorly by considering Autodoc Vina as our uh, docking tool. So this is more or less most of us are familiar. We have two types with the structure-based drug designing or ligand-based drug designing. Docking plays an extremely important tool, irrespective of the database which we consider. We may take from zinc or drug bank, Campbell or whatever. We may take 100 or 500 or 5,000 or 5 lakh molecules. And protein also, whether we may take from homology modeling or crystallography. But one of the crucial steps that we do in virtual screening is the docking. So this is one of the important aspects. And usually what is done is when we have, this is just one small example which I thought of using to demonstrate how our tool or the method that we developed becomes an important aspect. So one of the example that I took to demonstrate the tool is by using one of the protein called FFAR2, free fatty acid receptor 2, and this is one of the key enzyme required and which helps us in neurophysiology and immune function, pancreatic beta hemostasis, many things. So in this study, since we are not in this presentation, I am not more towards the biological aspect, but more towards the computational aspect. So in any study, usually the scientists, when we are working, we take like 1 lakh molecule or 2 lakh molecules, 1 million, 1.5 million, or zero point 0.5 million like this. So here like we have nearly zero point 0.1 million molecules from different databases, which are from the natural products. After considering this and after performing the docking, based on this Venus score, one may sort it out and then say like these may be the top 15 ligands. Here I have considered like the top 14, 15 ligands. And this needs to go for the next wet lab, either in vivo or in vitro analysis. Now, the major challenge that comes is, should we go with the first one, something which is having like minus 6.6 or minus 6.5 or minus 6.1 or minus 6? Now, if we critically observe this particular situation, there is no much difference between this and this, which is zero point 0.1 difference. And compared to this one, this is 0.4 difference. Now, both the molecules are having the same Vina score, minus 6.1. Again, minus 6.1 and minus 6 is not no significant difference. When here, if we look into, both are having the same, here all three are having the same. So which one is our candidate to go for the next wet lab analysis? Now, one of the method which typically as a computational chemist we do is we go for generating the Ligplots and then look at the hydrogen and hydrophobic interactions. And then based on the interaction, if the ligand is having a preferred hydrogen bond with a specific amino acid, then we consider that and go for the next analysis. Now, in the whole process, if we analyze, let's say in this case alone, is this example I have given to understand the complexity if we do this task manually. So I have 16 ligands here, and whenever we do docking with the Autodock Vina, it generates 9 conformations per ligand. We can change the number of conformations, but usually used is 9. So we have like 16 ligands into 9 docked conformations. So we get around 144 conformations now. Now to generate these 144 conformations, even if we take, let's say 2 minutes for one conformation to generate, it takes around 288 minutes to generate the Ligplots for all 16 ligands. This is only 16 ligands, but we usually have like when the case is like 144 um, 144,000 14400 ligands, so the time taken will be more, and the cases of getting the similar Vina score is also high. So if we do it manually, it takes around 5 hours to generate the Lig plots. And what we have developed is a method by which we can get the Lig plots, and also we will get the hydrogen bond and hydrophobic interactions within 20 to 30 minutes in max for these 16 ligands. So how does it work? So we have developed a method called Vinaliggen. So Vinaliggen refers to Vina is for Autodock Vina, and results to Ligplot generation. We generate the Ligplots from these Vina results. That is what this Vinaliggen refers to. So it extracts the hydrogen and hydrophobic interactions. It generates the PostScript files, and it considers the out.pdbqt, whatever we generate from the Vina, Autodoc Vina, and we need to input the macromolecule in the PDB format. Then it extracts the interaction data, and then we can also provide the shortlisted docked complexes. What is the shortlist docked complexes refers to is, let us say we have 15, 16 ligands here. I do not want to see all the interactions. Let us say only 3 ligands. I want to see the interaction. Then we can just provide the zinc score of 3 ligands of our interest. That is also possible in the method that is developed. Then we will have the tool which does not require any pre-processing. We can provide the zip file, and it works fine in generating the results. So we have made it like a 3 situations. Usually when we do the docking, either we may get this out.pdbqt directly, or like this one, or sometimes the file will be just given with the name, and the zinc ID will be within that file name if you open, or sometimes it goes within the folder, and the file goes within the folder, and if you open that file, then the zinc ID will be there. So we can choose any of these 3 ways whenever we are docking. Usually the method whenever we do, we get the results with the type 1. Like the zinc ID will be there, and then the minimization, if it is done through PYRX, so universal force field, and then we will also have the out.pdbqt. So this is how the page looks like, where we can upload our out.pdbqt files here. We can upload our macromolecule here. Like I have just put an example like ligands.zip. We can put all our docking result files here. We can upload all our PDB file, one protein PDB file here. And then shortlisted means whatever the ligands of our interest, I want to see only the Lig plots for those things. That is also we can put. Then we can say upload. Then since the file name is there on the um, front. So since it is not there within any of the folders, so we can select type 1, and then we can start. Then the processing of the ligand will happen. It will split the files, make the complexes, use the Lig plot, and then starts to generate the Lig plots for each of the conformations. So let me just quickly demonstrate how it works. And in the meantime, when it is running, I will just continue with this particular slide. So I hope this window is also visible.
Yes, it's okay.
Okay, so we can open in the terminal and then say Streamlit, and we have a file called main.py, and then it will open in the browser. So we don't need any internet connection also. Since if we have a molecules we thought of patenting or anything, so we can run it without connecting to internet. So we can just run it using this program. So we can upload the molecules here. So we can upload the protein here. We can upload the ligand complexes here, protein here, and we have this shortlisted. So I have given like only for these three ligands to generate the Ligplots, rather since we have a time constraint, so that we can just see how it works. So rather than 16, I have given like three ligands to process. Once this is done, we can say upload. Now everything is uploaded. Then we can say start. So it's running over here. And then we will also get to see the progress of the program over here. Now the splitting of the files is happening. It checks whether the Ligplot is correctly there or not. The Ligplot, hydrogen bond, hydrophobic plus.exes are there or not. So since we have uploaded three ligands, so each ligand will have 9 conformations. So 9 into 3 is 27. So now the ligand first one, which is labeled as 175, which is from the PubChem. So it's splitting and then making the Lig plot is happening. Then it takes the second ligand, it takes the third ligand, like this, it will be happening. So this process may take around 2 minutes, and then we'll get the final results. So in the meantime, I will just quickly finish with this one. So this is what is submitted now. We have uploaded, and then it is progressing. Once this is done, we will have the files created like this. So Ligplots will be there within this folder. Target files will have all the PDB files. Then we will have one file called output.csv, which will have for each conformation the hydrogen and hydrophobic interactions present within this. If we open any of the Ligplot, we will also get the postscript of the Ligplot, and we can also get the PNG image of the Ligplot. Now the question comes, why is this hydrogen bond and hydrophobic interactions are so important? More or less we are familiar. Why is this one required? Because hydrogen bond brings the molecular recognition, it brings the binding affinity, it makes the orientation of the ligand, it brings this is most important, this brings the specificity, it adds a lot towards the overall scoring function, helps in the drug discovery for optimization of the structures, and it has a major role in biological function and in the virtual screening. Same with the hydrophobic interactions, they bring the complementarity, it improves the binding affinity, and it looks for the non-polar region interaction also we will get to know. And interactions usually happens at the core of protein ligand complexes, core binding. This also helps in specificity and improves the binding strength and brings the structural stability. So these are the advantages. Why are we considering? Now, if we look at the result, anyway, I will also open the same result once the complex creation or Ligplot generation is over. So quickly to show, so once we have the simulation, once the command line is done, so this is the first ligand, 9 conformations, and along with this, the first conformation formed one hydrogen bond with the histidine 242. Now, why is this important is we will get a holistic view of one ligand and its corresponding hydrogen and hydrophobic interactions. So this ligand possibility of having a hydrogen bond is not there in all 9 conformations. Only in one conform or 2 conformations it is there, and it prefers to bind with histidine 242 with the bond distance of this 2.88 or 3.24. And other hydrophobic interactions are also possible with this particular ligand. That is also we are going to get to see. The second ligand is having more hydrogen bonds compared to the first one. And we can also analyze, like in most of the cases, it prefers to bond with this glutamic 166 in most of the cases. So we will get some view like what is its preference to. Then the third ligand over here, again 9 conformation, each conformation, and how is the hydrogen bonding pattern is, and wherein all the hydrophobic interaction is happening. So by looking into this uh, understanding of the ligand interaction with the protein, we can make some guess whenever we are doing with virtual screening, some guess that yes, it prefers to bind like this, and it won't interact with our pocket. It interacts with this pocket. So if we have those pocket information, selection becomes very easy. We will also get the PNG image, and we will also make the guess. So here is in one case where I did uh, the intuition based study. I said like this is anyway the potential agonist for that one. This is another ligand having like 70% chance that it acts like an agonist, and this is another ligand which acts like a 50% as agonist based on their binding pattern. And then our collaborator tested these molecules, and then we got to know like this penchol, which is from the basin plant, gave a very good result. So from 1,44,000 ligands after screening, after now, if this 81099, if we look into back to this one, this is the one 81099, which is relatively less in the binding energy compared to this. Now, if we just go with the energy and then finalize or go for the wet lab, we would have missed this molecule. So because we have this interaction details, so we are in a better position to decide which one is good. So now if I go back to this, now this is done, and it took around 2 minutes 27 seconds to complete the overall Ligplot generation, and we can open this and we can have this output dot CSV. Now this output dot CSV, we can go for understanding like yes, this is where the hydrogen bonding is happening, and this is what the more hydrogen bond, this is a less hydrogen bond, and again goes with what is the preference or what is the project is. If some ligand wants to have 2 hydrogen bonds with 2 different amino acids, that can be picked up. So if there are any questions, I will be more than happy.
Okay.
Yes, sir. How do you screen agonist from antagonist?
Well, from the docking, it is always difficult to decide like agonist or antagonist. But in our case, what we did is since the acetate, butyrate, and propionate all are the agonists, which when they bind to the protein, they're activating. So we made an attempt to understand their interaction profile. So how exactly the existing agonists interact? So these molecules actually forms 1 hydrogen bond either with arginine 180, arginine 255, and supported in the back with the histidine 242, not in case of antagonist. So based on the pattern, how the existing agonists interact, and then we made an analysis like yes, my our ligand of interest is also forming the similar kind of a interaction, so it may act like an agonist. Again, wet lab analysis is required, If this information is then it is difficult.
Okay, thank you. I see 2 questions in the chat, but the time is over. So probably you can answer via chat or via email to Vladimir Sulimov. He has 2 questions about hydrogen bonds energy estimations.
Okay, I will look into that. Yeah,
Yeah.
thank you.